Hello friends, we will study active transport in this video. Cells sometimes absorb materials from outside environment, although the concentration of this material inside the cell is higher than outside. It means cell material concentration in cytoplasm, for example 10, but in environment 5. Unlike diffusion, that time materials move from low concentration to high concentration. To perform this process, there must be carriers in cell membrane. If we recall the structure of cell membrane, we said it consists of two layers of phospholipids, and between this layer of phospholipids there are carrier proteins to transport material inside and outside of cell. These carriers can move inside, outside the cell. The carriers integrates with other material, particle or ion needed by cell. Then this carrier moves to inner surface of the membrane, left the material and go back to outside. Like from that side, a protein capture some particle, move inside, left in cytoplasm, after go back to the surface again. But this process requires energy. As we see, carrier proteins are active, but this action need energy, which is supplied by ATP. Active transport mainly question to ask about it before they ask about how to draw active transport, my friends. But we need to remember structure of plasma membrane. Just we draw it and draw some material outside and more than this inside, and show the way or direction of material transport from low concentration to high concentration to illustrate the active transport. Another question is comparison between active transport and diffusion. So you remember we said in diffusion materials move from high concentrated medium to low concentrated medium, while in active transport material moves from low concentrated medium to high concentrated medium. In diffusion, energy not used, but in active transport energy, ATP energy is used. In diffusion, proteins or carrier proteins in plasma membrane, they are not active, but in active transport, these carriers are active, integrate or carry materials or ions from outside to inside or from inside to outside. There are three main differences between active transport and diffusion. The phagocytosis, pinocytosis, and exocytosis, they label it as different or not part of active transport, but in that, as a question asked to give examples for active transport, we can give phagocytosis, pinocytosis, and exocytosis. All these three types of material passage through plasma membrane are examples for active transport. Phagocytosis is cellular eating. It is common way of eating or nutrition among protista like amoeba. You may remember from previous years, amoeba, an organism which can change its shape. What meat out a solid food particle, it forms the pseudopodia, surround the food, take into the cytoplasm and digest it. It also used by white blood cell devouring remaining cells and germs in the blood. As you remember again from lysosome, we said neutrophils, which kind of white blood cells, they reach in lysosome because they have function of phagocytosis in devouring means phagocyting or eating germs, bacteria. This process is done when cell membrane make a pouch-like structure that surround the food, solid matter. Then this part separate from the cell surface and moves inside the cytoplasm like this, food inside. The contents are digested by the enzyme and secreted by lysosome in the cytoplasm. We remember lysosome contain enzymes which their function is digestion. For pinocytosis, it is similar to phagocytosis, means mechanism is similar, but that time the organism meet with a liquid matter but not solid. When liquid material is taken, there will be a small hole in the plasma membrane. This hole again surrounds the matter and becomes a pinocytic vesicle. And the structure which contains food in cytoplasm or store it until digestion, then we call it pinocytic vesicle. And what about phagocytosis? This vesicle, we can call it temporary food vacuole. Why temporary food vacuole? You may remember from animal cells. Because it is temporary, it stores this food until it being digested. After food digests, the useful material diffuse into the cytoplasm, but wasted are discharged out of cell by exocytosis. In that way, we call it temporary food vacuole, or we can call it also phagocytic vesicle. The main question is asked about phagocytosis and pinocytosis is comparison between them or writing the differences. Phagocytosis, cellular eating, while a solid food particle is captured by cell. In pinocytosis, but a liquid material is captured by cell. In phagocytosis, we can say temporary food vacuole formed or phagocytic vesicle is formed, but in pinocytosis, pinocytic vesicle is formed. In both of them, they fuse with lysosome to digest this particle and after release the material out of cell by exocytosis. The exocytosis term is used to describe the release of some materials outside the cell. It represents the excretion in plants or animal. This process takes place in various cells to get rid of undigested leftovers from the phagocytosis or secretion of some hormones. We not mainly focus on that part because mainly hormone discharging from cell, we call it secretion, but undigested food material, removing them from cytoplasm, we can call it exocytosis. And it's eating of cell, phagocytosis, drinking in cell, pinocytosis, excretion in cell, exocytosis, and they all examples for cell metabolism. Cell metabolism is represented by some of all chemical changes happen in cell which takes place with the help of enzymes. So any reaction in cell need a different enzyme. These changes are catabolism, which is degradation with digestion of material, and anabolism, which means building new things. Degradation, catabolism, building, anabolism. Anabolism usually consumes energy, like while catabolism release energy. Anabolism and catabolism. Because question about this paragraph can ask you about definition of cell metabolism, which we can say all the chemical changes happen in cell are called at metabolism. Or catabolism, the chemical changes happen in cell which broke down big molecules into smaller particles. Big molecules are broken down into smaller, while in anabolism small molecules are used to build complex molecules. In anabolism, it consumes energy or use energy. Like ribosome need energy to build or to produce proteins. Smooth endoplasmic reticulum need energy to build fats or complex carbohydrates. Or we all know about this chloroplast need sunlight energy to produce glucose, organic material. They all anabolic reactions consume energy, while catabolic reactions release energy. Example for catabolic reactions is mitochondria and cellular respiration, while glucose broke down into smaller pieces and ATP energy released. That's why we call mitochondria is powerhouse of cell, or main function of mitochondria is cellular respiration, in which ATP energy is produced. Glucose particles are anabolized from small CO2 and small H2O to form bigger molecules, glucose. So example for anabolism. Green plants obtain this energy from the sun to prove that the anabolic reaction uses energy, while catabolism of glucose particle is respiration, which aim is releasing the energy, ATP energy, by the living organisms to be used in many activities. One more we can write as organic anabolism or photosynthesis is example for anabolic reaction, and for catabolic reaction is cellular respiration. In our next lesson, we'll continue with respiration.